Hey science enthusiasts, welcome back to another exciting dive into the world of molecular biology. Ever wondered how scientists make millions of copies of DNA in just a few hours? It's like magic, but with science. Meet polymerase chain reaction, or PCR, the ultimate DNA photocopier. This technique revolutionized genetics and molecular biology. Alright, let's break it down with the three-step dance that PCR performs. First up, denaturation. At a scorching 95 degrees Celsius, DNA says, "I need space," and splits apart into single strands. This is the first crucial step. Imagine a pair of tightly coiled hair ties suddenly snapping apart. That's your DNA during denaturation. Next, we cool things down to 65 degrees for annealing. This is where the magic starts to happen. Here, short primers. Think of them as tiny DNA helpers. Stick to the template strand like puzzle pieces. They are essential for the next step. It's like a matchmaking party for DNA strands. The primers find their perfect match and bind to the DNA. Finally, extension at 72 degrees Celsius. This is where the real action happens. This is where DNA polymerase gets to work, building new DNA strands like a tiny, overworked factory worker. It's a fascinating process. It's like a minuscule assembly line, churning out perfect copies. Each cycle doubles the amount of DNA. Repeat this cycle 30, 40 times, and boom, you've got billions of copies of your DNA. It's truly mind-blowing. But wait, there's more. Let's talk about different types of PCR techniques. Each has its unique twist. First, conventional PCR, the OG method, perfect for basic DNA amplification. It's the foundation of all PCR techniques. Then we have qPCR, or real-time PCR. This one detects and quantifies DNA while it's being copied. Fancy, right? It's incredibly useful for precise measurements and diagnostics. Next up, RT-PCR, or reverse transcriptase PCR. This technique converts RNA to DNA before amplification. You've probably heard of it from COVID-19 testing. It's a crucial tool in virology and disease research. Multiplex PCR, this one's for the multitaskers. It detects multiple targets in a single reaction, because who has time for one at a time? It's like juggling multiple balls at once, but with DNA. And finally, digital PCR, like qPCR, but more precise and perfect for detecting tiny amounts of DNA. It's the future of DNA quantification. Now let's lighten things up with some funny PCR fails scientists know too well, because even in science, things don't always go as planned. Forget to add Taq polymerase? Congrats, you just made water. It's a classic rookie mistake. Got contamination? Oops, you just PCR'd yourself instead of your sample. Contamination is every scientist's nightmare. Ran out of DNA? Time to pray to the PCR gods. Sometimes you just need a little luck. If you're loving this microbiology content, make sure to follow at World of Microbes for more fun and informative science videos. We've got plenty more to share. And hey, drop a comment if you've ever had a PCR fail, or tell us which technique you want to learn next. We love hearing from you. Stay curious and keep exploring. The world of science is vast and full of wonders. See you in the next video. Until then, happy experimenting.